Bismillah ar-Rahman ar-Rahim. This is Professor Ahmad Alsaad. I will discuss in this presentation the physical aspects of the nervous system. This presentation is very important for students who are going to be specialized in the nervous system of our bodies, the neurology. In the first slide, I discuss the neuron cell, which is the main component of our nervous system. The neurons, also called the neurons or nerve cells, are the fundamental units of the brain and nervous system. These cells are responsible for receiving sensory input from the external world and then sending motor commands to other organs of our body, and also they are responsible for transforming and relaying the electrical signal at every step in between. The neuron cell sends the command from the brain to our organs of our body to respond to these commands. Every action that we do, every activity that our body does receives the commands from the brain, from the nervous cells in the brain. In order to understand the functioning of our nervous system, we need to understand the nerve conduction. The information is transmitted in the human body by electrical pulses in nerve fibers called axon. Axon is a very important component of our nervous system. It transmits the nerve pulses from the neurons to the other organs of our body. An axon can be thought of as a conductor that has a very high resistance, and this conductor is filled with a conducting fluid called exoplasm. In fact, the resistance and the resistivity of the conducting fluid, the exoplasm, is very high. Now, this conductor is poorly insulated from the surroundings by a thin, flat membrane that is made of a very bad insulating material. This is why most of the nerve current in the axon leaks out from the membrane, through the membrane, to the surrounding interstitial fluid in a very short distance. This is why the nerve pulses in our body need to be amplified many times before this nerve pulse can reach the targeted organ of our body. After a very short distance, nerve pulses become very weak. Why they become very weak? Because most of these nerve pulses leak out through the membrane to the surrounding interstitial fluid, and then they must be amplified. We will talk about this later on when we talk about the two types of the axon. It was found that the speed of the nerve pulse is just about 1/1,000,000 of that of the electric pulse in a wire. Why is the speed of the nerve pulse is very small? Because as I told you, the axon is coated with a membrane, and this membrane is a very bad insulator. It has a resistance that is very, very, very small in comparison with the resistance of the axoplasm in the axon. Let us now discuss the structure and functioning of the neuron. Neurons, also known as nerve cells, send and receive signals from your brain, and while neurons have a lot in common with other types of the cells in our body, they are structurally and functionally unique, which means that the function and the structure of the nerve cells in our brain and in our nervous system have different functioning and the structure than other cells in our body. As you see, the main component of our nervous system is the cell body, the neuron or the nerve cell, and this nerve cell is connected to other parts of the nervous system and other neurons by a long thin conductor called an axon. This axon could be as short as 1 centimeter or as long as 1 meter in our body, and this depends on the function of the axon, okay, and what types of the nerve pulses it transmits. These axon usually transmit the electrical and chemical signals from one nerve cell to other nerve cells in our nervous system. As you have seen in the previous slide, you saw that there are several components of the nerve cell or the neurons. The primary components of the neurons are the cell body, which is called soma, then the axon, and the axon is a long cylinder projection that conducts electrical and chemical impulses away from the cell body to the other parts of our nervous systems. And then we have what's called dendrites. They are tree-like structures that receive messages from other neurons, okay, and then we have what is called the synapses, specialized junctions between neurons. You don't have really to memorize the components of the neuron. All you need to do is to understand the function of the axon, because here we focus only on the electrical properties of the axon in our nervous system. As I mentioned in a previous slide, that the electrical aspects of the axon are really what matter in this course. For axon, we really need to understand two types of electrical potentials. One is called the resting potential. This occurs when the axon is undisturbed, which means that the axon is not functioning at all, or there is no nerve impulse to stimulate the axon. So the interior of an axon is at lower potential than the surrounding interstitial fluid. So, and in this case, there is no nerve impulse is being transmitted. While if a nerve impulse stimulates the axon, okay, then we should define what is called an action potential. And in this case, a current pulse travels along the axon. A nerve current impulse travels along the axon. In order to understand the electrical aspects of the axon, we need to understand that the axon can be thought of as an electrical long cable that contains a conducting fluid called axoplasm with a very high resistivity and resistance, and this conductor is coated with a deductive insulation material or a very bad insulator which has a resistance that is much smaller than the resistance of the conducting fluid in the axon. This insulator is in the form of a thin flat membrane that allows the current to leak out to the surrounding interstitial fluids in many places, because its resistance is really much smaller than the resistance of the axoplasm in the axon. In the next slide, I will show you the model that we use in order to understand the electrical aspects of the axon. So the axon consists of a cylindrical membrane containing a conducting fluid, the axoplasm. The current can travel along the axon in this fluid, and because the resistance of the membrane is smaller than the resistance of the axoplasm, it leaks out through the membrane, and then in order for this nerve impulse to survive, it should be amplified many times before it can reach the targeted organ. As we have seen in the previous slide, the axon can be visualized as a cable, which is a conductor that contains a conducting fluid called an axoplasm. This cable is cylindrical, so it has a length h and has a circular cross section of radius r, and the resistance of the conducting fluid axoplasm is r. And when this axon is stimulated, okay, then the nerve pulses are transmitted along this cable, and they form a nerve current called I axon. I axon then represents the nerve impulses being transmitted in the axon, and R is the resistance to the flow of the nerve impulses. In other words, it's the resistance to the I axon. Now the conducting axoplasm has a resistance R to the current I axon along the axon. Now, as we mentioned before, this cable is coated with a deductive insulation in the form of a flat, thin cylindrical membrane that surrounds the axon. Now, if we take a unit area of this membrane, then we have to define what is called a resistance of a unit area of the membrane, Rm. M means membrane, so this is the resistance of the membrane, and this resistance is much smaller than R. Okay, so this is why most of the I axon leaks out through the membrane, and the leakage current is called I leak. I leak means the nerve impulses that leak out from the axon through the membrane to the surrounding interstitial fluid of our nerve system. Now, this Rm is a constant parameter, and you do not need to memorize it. It will be given to you. It depends on the type of the axon that we are dealing with. There are two types of the axon. One is called myelinated axon, and the other is called unmyelinated axon. And this Rm is different from myelinated to unmyelinated axons. And in the problem given to you, Rm will be given to you as a constant parameter. In this slide, I focus on the structure and functioning of the membrane. As I mentioned before, the membrane can be visualized as a thin, flat, cylindrical membrane which has a resistance that is much smaller than the resistance of the axoplasm, and since the charges can accumulate on the two sides of the membrane, the interior and the exterior of the membrane, this means that we can think of the membrane as a parallel plate capacitor with two metal plates. One is charged with a positive charge and the other is charged with a negative charge, and of course an electric potential difference is created between the two plates, and an electric field is being generated so that the nerve impulses can be stimulated by this electric field, and then they can leak out from the membrane to the surrounding interstitial fluid. So here we will define a constant parameter called Cm. This resembles the resistance of a unit area that we defined in the previous lecture, but this Cm is defined as the capacitance per unit area. The unit is farad per meter squared, while the unit of Rm is ohm meter squared. This Cm, the capacitance per unit area of the membrane, is defined as the charge per unit area, which is the surface charge density, divided by the resulting electric potential difference V. And the unit of Cm is again farad per meter squared, and it will be given to you in the problem as a constant parameter. In order to appreciate the electrical properties of an axon, we will now calculate resistance of the axon. Then we will calculate the capacitance of the membrane, and then we will calculate the resistance to the leakage current through the membrane. In order to calculate the resistance of the axon, as you know, the axon can be visualized as a long conducting cylinder of length h and radius r that contains the axoplasm of resistivity rho a. Since the resistance is directly proportional to the length and inversely proportional to the cross-sectional area, and the constant of a proportionality is the resistivity, so the resistance of the axon R equals rho A h over pi r squared. Rho A is the resistivity of the axoplasm, h is the length of the axon, and r is the radius of the cross-sectional area of the axon. Now, as we mentioned before, there are two types of axon. One is called myelinated and the other is called unmyelinated axon. For a myelinated axon of length 1 centimeter, and if the axoplasm resistivity rho A is 2 ohm meter and the radius of the axon is 5 microns, 5 micrometer, then the resistance of the axon R, this is the resistance R to the current I axon, is 2 ohm meter times the length 0.01 over pi times r squared, and then you will get here 2.5 times 10 to 8 ohms. We here forgot to square the radius, but all you need to do is to square the radius. This should be squared. In this slide, I show you how to calculate the capacitance of the membrane, the thin, flat membrane surrounding the axon. So, since the membrane is quite thin, so a small section appears nearly flat. So the membrane can be visualized as a per plate capacitor of length h and the cross-sectional area A. So a length h of the membrane has a surface area because it is cylindrical, so the surface area A is 2 pi r h, where r is the radius of the axon and h is the length of the axon. And since the capacitance per unit area of the membrane is Cm, so the capacitance of length h of the axon C equals Cm times 2 pi rh. Let us take an example, a numerical example. Find the capacitance of 1 centimeter length of an unmyelinated axon of radius 5 micrometer, where the capacitance per unit area for the unmyelinated axon Cm is 10 to minus 2 farad per meter square. If you plug in the numbers, you will get that C equals 10 to minus 2 times 2 times 3.14 times 5 times 10 to minus 6 times the length 0.01. Then you will get 3.1 times 10 to minus 9 farad. This is equivalent to 3.1 nanofarad. This is the capacitance of unmyelinated axon of a length 1 centimeter and a radius 5 microns. Let us now see what would be the value of the capacitance of a myelinated axon for the same length 1 centimeter of a myelinated axon and the same radius 5 microns. The capacitance per unit area of the membrane in this case is given to you. Cm equals 5 times 10 to minus 5 farad per meter squared. See here that Cm for myelinated axon is much smaller than the Cm for unmyelinated axon. So if we calculate C in this case, we will get 6.28 times 10 to minus 12 farad. We will get then 6.28 picofarad. This capacitance is about 200 times smaller than that of the unmyelinated axon. The third parameter that is important to be calculated is the leakage resistance. Leakage resistance represents the resistance to the I leak, to the current leaked out from the interior of the axon through the membrane to the surrounding interstitial fluid. Since the membrane is not a perfect insulator, we mentioned that the membrane is defective insulator, bad insulator. Its resistance is much smaller than the resistance of the axoplasm. Charge, we mean here by charge, we mean nerve impulses, leaks from the axoplasm through the membrane into the interstitial fluid. The resistance of a conductor, as you know, is directly proportional to the length and inversely proportional to the cross-sectional area. So if the resistance to leak currents through a unit surface area of the membrane is Rm, and Rm will be given to you in the unit of ohm meter squared, then a portion of the membrane with surface area A has a resistance R prime equals Rm over 2 pi Rh. Why we use 2 pi Rh? Because the membrane is cylindrical. So this is the surface area of the membrane. Now find the leakage resistance of a membrane of unmyelinated axon of length h equal 1 centimeter and resistance of a unit area of the membrane Rm is 0.2 ohm meter squared. So R prime equals Rm over 2 pi Rh. So it's 0.2 over 2 times 3.14 times 5 10 to minus 6 times 0.01. So in this case you will get 6 times 10 to 5 ohm, which is less than 1% of the axoplasm resistance that we calculated and found it to be 2.5 times 10 to 8 ohm. Since this resistance is very small in comparison with the resistance of the axoplasm, most of the current entering an axon segment leaks out through the membrane in much less than 1 centimeter. In order to calculate the distance by how much distance the nerve current can travel in the axon before it leaks out through the membrane, we need to find what is called a space parameter. A space parameter lambda is a distance that measures how far a current travels before most of it leaked out through the membrane. Now, as we calculated before, the resistance of the axoplasm is R equals rho A H over pi R squared. As you see from this formula, that R is directly proportional to H, where H is the length of the axon. The leakage current suffers from a resistance called leak resistance R prime, and R prime equals Rm over 2 by H, where Rm is the resistance of a unit area of the membrane. As you see here, the leak resistance R prime is inversely proportional to H, and this is why there is some distance called lambda, space parameter, for which the two resistances R and R prime are equal. So when at a given distance R increases linearly with H and R prime decreases linearly with H, this means that there is a distance called lambda at which R and R prime become equal. So just put lambda for h in the formula of r and r prime, then equate r with r primes. So rho a lambda over pi r squared equals rm over 2 pi r lambda, and from here you can find that lambda equals the square root of rm r over 2 rho a. So this lambda is constant for a given axon, because rm is constant, and if we take an axon with a specified radius, the resistivity rho a of the axoplasm is given, then easily you can calculate what is called the space parameter. Again, we will focus that this lambda measures how far a current it travels before most of it leaked out through the membrane. For unmyelinated axon, we know that Rm is 0.2 ohm meter squared, the radius is 5 microns, and the resistivity of the axoplasm is 2 ohm meter. And if you plug in the numbers, you will find that lambda is 5 times 10 to minus 4 meter, 0.5 millimeter. This means that the nerve impulse, it travels only 0.5 millimeter in the unmyelinated axon before it leaks out through the membrane to the surrounding interstitial fluid. This is why every 0.5 millimeter this nerve impulse should be amplified to survive and to continue traveling through the axoplasm until it reaches the targeted organ. For myelinated axon, if we calculate lambda, the only difference between the unmyelinated and myelinated axon is in the value of Rm. Rm is much larger for myelinated axon. It is 40 ohm meter squared. So if you plug in the number, of course we are taking the same radius, 5 micron, and the resistivity of the axoplasm is the same, 2 ohm meter. And if you plug in the numbers in the formula, you will get lambda for myelinated axon 7 times 10 to minus 3 meter, which is a 7 millimeter. This means that in the myelinated axon, the nerve impulse can travel 7 millimeter, see, less than 1 centimeter, before it leaks out through the membrane to the surrounding interstitial fluid. So every 7 millimeter, the nerve impulse should be amplified to survive and to continue traveling along the myelinated axon so that it can reach the targeted organ.